This video is all about the action potential. How, at a cellular level, do neurons initiate an action potential? And then once it's initiated, how does that action potential propagate all the way to the end of the nerve cell where it can cause transmitter release? Previously, I talked about an experiment and I said we could inject current into a cell. And if we injected enough positive current into a nerve cell, what would happen would be that that cell would reach threshold and the cell would initiate an action potential at the axon hillock. Also remember that based on the equilibrium potentials for sodium and for potassium, we could guess that the rising phase of the action potential is due to an influx of sodium into the cell, while the falling phase is due to the exit of potassium ions from the cell. So in this video, we got to talk about how ions actually move across cell membranes and then come back to this action potential. So remember that lipid bilayers are not permeable to ions. There's a gradient for sodium to move into the cell, but it can't do it. There's an electrochemical gradient for potassium to move out of the cell, but potassium by itself can't pass through the lipid bilayer. And so in biological membranes, there are helper proteins that will allow this movement. These proteins are called ion channels. There are a lot of different types of ion channels, over 100, but we can talk about them generally here. So ion channels are going to be membrane proteins, and they're going to form pores that allow ions to move. They're selective for a particular type or types of ions, so there'll be some portion of the protein that will define a selectivity filter. And what I mean by that is that in a sodium channel, there'll be components of the protein that will allow only sodium ions to move through, but will make it unlikely that a potassium ion or some other kind of ion will move through. And similarly, potassium channels will exclude chloride ions and things like that. Ion channels are also gated so that they act like doors. They're not open all the time. They can either be closed or open, and they'll be a part of the protein that will act as a physical barrier that will prevent movement through the pore. We call this the gate. Now sometimes, but not always, ion channels also contain an activation domain, and I'll talk about that in a little bit. Let's look at voltage-gated sodium channels. This is the key player in an action potential initiation, and they're going to be sodium channels that are going to open when the voltage changes. Sodium channels can exist in 3 states. Right now, what I've drawn here is a closed channel. You can see that the gate is blocking the flow of ions through the channel, so that even though there's an electrochemical gradient for sodium, it can't move through the passage. What will open this type of channel is if the cell becomes more positive. That's why we call them voltage gated. So in response to the cell getting more positive, now the voltage gate slides out of the way, the channel's in a new configuration, and sodium can move through the pore and enter the cell, depolarizing it. But voltage-gated sodium channels also have an inactivation gate, and what that means is that even if the cell stays depolarized and the voltage gate's out of the way, the inactivation gate will adopt the conformation where it will block movement through the channel. And now, again, despite the gradient, sodium will no longer be able to move. The switching between closed to open to inactivation is very rapid. It takes fractions of a second, fractions of a millisecond to make these changes. Neuron membranes also contain voltage-gated potassium channels. They're a more diverse group than voltage-gated sodium channels, and so again, I'll talk about them generally, but there'll be differences. Like voltage-gated sodium channels, they can exist in closed and open states. So here I've drawn a potassium channel that's closed. The pore is blocked by the gate, and thus, even though there's an electrochemical gradient for potassium, can't leave. They will open in response to an increase in voltage. So if the cell becomes more positive, then the voltage gate slides out of the way, and now potassium can move down its electrochemical gradient, and it would leave the cell. Some potassium channels have no inactivation gate, and so they could only exist in these 2 states. Others will rapidly inactivate, like the sodium channels, and there are still others that will inactivate very slowly. When the channel inactivates, it's similar to what we've seen with the voltage-gated sodium channel, where there'll be another part of the protein that will slide in, blocking the pore, and then potassium cannot move through. All right, Well, our goal is to explain how we get an action potential. Let's go ahead and look at these events in the axon initial segment, see how these channels play. So I've drawn a blow up here of the very first part of the axon. The cell body would be somewhere off to the left, and then we have the axon here. And what we're looking at in that very part on the left would be the hillock region of the axon. It would be also called the axon initial segment. And as my cartoon shows, the axon initial segment contains many voltage-gated ion channels, so they're highly concentrated there. Now this cartoon shows us something else, right? It shows us that the axon's not uniform. So moving over here, you'll see that there are also these other segments where we got a lot of ion channels. These are called the nodes of Ranvier. Nodes of Ranvier are these short gaps in the axon. They're on the order of about 1 micron long, so not very wide at all. And they're bare, meaning that there's not myelin sheath wrapped around them. And in these spots along the axon, there's a high concentration of membrane proteins, including voltage-gated ion channels and the sodium-potassium ATPase pump. Nodes of Ranvier are separated from one another by things called the internode region, shown here. And this is the part of the axon that will be wrapped by glial cells, or myelin sheath. Myelin sheath varies in thickness from different kinds of neurons. Sometimes it's not present at all, but when it is, it can be between 0.5 and 5 microns thick. And the internodes themselves are really quite long. They can be up to 1.5 millimeters in length, so like 1,500 times longer than the nodes of Ranvier. So I'll draw this little separation here just to have you remember that I've not drawn the internodes to scale. In the internode region, there are a few membrane proteins. All right, let's look at the action potential and see what happens, beginning with what's going on in the resting phase. So when the cell is at rest, there is no action potential happening, and the reason for this is because the voltage gates are closed. The voltage gates are closed, nothing can move, no ions are moving back and forth, it's resting. Now, what happens if we depolarize the cell by injecting current or by having the cell get more positive? Well, under those circumstances, positive ions will flow into the cell and the voltage gates open. When the voltage gates open, that allows sodium to move into the cells, which can open even more voltage-gated ion channels, and that can lead then to the rising phase of the action potential. Near the peak of the action potential, two things are happening. The first thing that happens is we see that the sodium influx slows as the cell approaches its equilibrium potential for sodium. And the reason for that is that at equilibrium, you'd have an equal number of sodium ions entering and leaving the cell. That's what equilibrium means. And so when we get close to there will be fewer ions moving back and forth. But in addition, a second thing's happen, and that's that the sodium channels are inactivating. And as they inactivate, no more sodium can move through. The falling phase of the action potential involves the potassium channels. So potassium channels are voltage gated, but they're slower to open than sodium channels, so it takes them a little bit, but then they'll open up. And once they open up, then potassium exits from the cell, and as it does that, it's going to push the cell interior down towards the V equilibrium for potassium. There's an undershoot phase, whereas the cell voltage falls, the voltage gates shut and they block ion flow. And then once the potassium channels are closed and the sodium channels are closed, the cell will return to the resting voltage, it's reset and ready to go, and it can fire another action potential. That raises the question then, well, how does an action potential initiated at the axon hillock propagate to the terminal? What does this mean at a cellular level? Well, let's think about this using an experiment. So suppose that I've got an axon and I can inject current in a spot, and then I can record the voltage at a particular spot some distance away. If I inject current at that one spot, well then sodium channels will open, the sodium comes in, and if it's sufficient to reach threshold, right, we'll initiate an action potential and I can record that. Well, suppose that I repeat this experiment, but now I've moved my recording voltage a little ways away. Once again, I've got my electrode, I can inject some current, but I'm recording further apart. Well, when I inject the current, sodium will enter and it will initiate an action potential there, but because my recording electrode's a little ways away, I don't see it yet. However, as that sodium is entered, it spreads laterally through the axon, and that positivity can open ion channels that are close to the recording electrode, and sodium will flow in there, initiating action potential. So at the spot of my recording voltage, now I see an action potential, but it's delayed relative to when I injected the current because it's some distance further, and we have to wait for channels to open and close for this to happen. If I had my recording electrode even further away, then what would happen would be sodium would come in and it would spread. Sodium would come in, it would spread, and finally we'd get to that set of voltage-gated ion channels near my recording electrode, and when the sodium entered there, we'd see the action potential, but again, with the time delay here. The speed that an action potential propagates along the length of the axon depends on a number of factors. Depends on the size of the axon. Axons can vary quite a bit. Some of them are quite small. They might be 0.3 microns in diameter, or they might be as large as 20 microns in diameter. It also depends on the degree of myelination, and there are some neurons in mammals that aren't myelinated at all, and others that are heavily myelinated. In fact, we can take different mammalian neurons and we can classify them into several groups depending on their size and myelination, and these different groups have different speeds of action potential propagation. We can see these differences when we look at a cross-section through a nerve. This is an electron micrograph, and here's a cross-section through a peripheral nerve taken from a rat. And pretty much all the cells that we see here are neurons, but they're not all uniform. So here, for example, is a cross-section through an axon of a single neuron, and you can see it's quite large. And all of these membranes that are surrounding it, this is the myelin sheath. So this is a neuron that's big and it's heavily myelinated, and that would represent a class of neurons that have the fastest speed of propagation. They might send action potentials as fast as 120 meters per second. Other axons in this image, the ones that I've outlined in blue, are quite a bit smaller, and you can see that they're not myelinated as much. These neurons might propagate action potentials only at half a meter per second. So there can be a big range of speeds in how fast the nervous system can process information, and it's largely related to myelination. We've now seen that we can initiate an action potential at the hillock. We know that we can propagate it along the length of the axon and get it to the terminal, whether it's a myelinated or an unmyelinated neuron. How then will transmitter be released at the terminal? Well, that's the topic for the next video.